Welcome to the Ask a Scientist video series. In this episode, we'll talk about using low-quality samples in NGS target enrichment. The quality of genomic DNA can greatly impact target enrichment sequencing workflows. Low-quality DNA samples are samples that are degraded or fragmented, damaged, contain contaminants, or contain a very low concentration of DNA. Common sources of low-quality DNA include formalin-fixed paraffin-embedded tissue, ancient samples, or circulating cell-free DNA. When samples are very low in concentration, they are considered low quality. This is often the case for cell-free DNA. When fewer copies of the genome are present in the input DNA sample, the final sequencing library will represent fewer genome equivalents, lowering the overall sample complexity. Additionally, low-quality DNA can be fragmented or contain damaged bases or regions resulting from sample age, processing, or storage. The original sample source can also be a factor. For example, DNA extracted from FFPE samples is often both damaged and low in concentration. Specific types of DNA damage and degradation include bases that have been oxidized or deaminated, abasic sites, and single or double-strand breaks. Low-quality DNA can lead to challenges in library construction and can yield lower quality libraries compared to libraries made with high-quality DNA. For example, libraries made using FFPE DNA often contain smaller insert sizes compared to libraries generated from high-quality DNA. Additionally, library conversion rates and PCR amplification efficiency can both be reduced when damaged DNA is used as input into library preparation. This leads to decreased library yields. Thus, to reach the library yields required for target enrichment workflows, additional rounds of PCR are often required, leading to increased PCR duplicates and reduced library complexity. Following target enrichment and sequencing, the impact of low-quality samples on several important sequencing metrics can be observed. For example, the need for more amplification increases the number of PCR-generated duplicate reads. When these duplicate reads are filtered from the data, the overall target coverage is decreased, and fewer genome equivalents are represented in the final data. Coverage uniformity is an important sequencing metric in NGS target enrichment because it measures how evenly each target region is covered. Low-quality samples can impact coverage uniformity due to poor library conversion, missing genomic regions as a result of DNA damage, or higher duplicate reads mapping to certain regions, all resulting in lower coverage compared to other targeted regions. There are several ways you can optimize your target enrichment workflow to improve the quality of your sequencing data. These include performing sample input QC and adjusting the sample prep workflow as needed, maximizing the input of degraded or damaged DNA samples, using high-quality library preparation reagents with high conversion rates, and using robust target enrichment reagents. Each of these steps can increase the quality of target enrichment sequencing data, enabling you to get the most information from every sample. Thanks for watching Ask a Scientist. If you have any questions or ideas for future topics, we would love to hear from you.